Origins was originally intended to only be a 3-part series, but we have decided to extend the show as new information on the origins of SARS-CoV-2 has come to light. If you are one of the thousands of people who have enjoyed this series, please consider giving us a small monetary donation to support our work. We have a Patreon account at patreon.com/media, as well as a Cash App account at DA Media 2021. That's $, D, A, Media 2021. Also, please rate and review this show on Apple Podcasts and share and suggest it to friends who you think would find it interesting. Thank you for listening.
I'm Senator Dr. Roger Marshall from the great state of Kansas. Before we dive into the story of the origins of COVID, questions I'm often asked are, why is this important? Why do we care about the COVID origins? There are many reasons why we need to get to the bottom of this virus origin. As a physician, I think we always need to know what, where, how, and perhaps why whenever any infectious disease outbreak occurs. We need to know the answers to the questions to prevent or minimize future infections, to develop future vaccines as well as antidotes. And if it was leaked from a lab, what can we do to prevent future leaks? And if it was made in a lab, certainly that would influence the height and thickness of those guardrails to do our best to make sure nothing like this ever happens again. 4 months prior to COVID landing in the United States, there's strong evidence to prove that China knew about the lab leak that began its coverage.
In China, the city of Wuhan is still on lockdown, with Americans trapped inside as the number of worldwide cases
in Italy has surged by 919 in a single day. That is
more than doubling.
Scientist Tulio de Oliveira discovered the new variant after observing a dramatic
is acting as an accelerant to conspiracy theories. The digital era we live in
came as the United States recorded more than 2,400 deaths in just
that has been reported from Brazil. And the third has come as the more contagious variant of COVID-19
the number of deaths recorded from COVID in the UK has
from a wet
passed
market in Wuhan, China, show it likely played a role in the development of
China's study into the origins of COVID-19. They've concluded that it's unlikely it came from a Wuhan lab.
I'm John Duffy, and this is Origins, Birth of a Pandemic, Part 5. The voice you heard at the beginning of this podcast was that of Senator Marshall of Kansas, who also happens to be a physician. Senator Marshall released what he called an 8-step plan to get to the origins of COVID and to hold China accountable. This plan includes some reasonable steps, including opening an investigation into the genomes held by the EcoHealth Alliance and an immediate moratorium on gain-of-function research. The plan also includes what appear to be more politically motivated actions, including sanctions and immigration restrictions on China. Senator Marshall released his video and 8-step plan after President Joe Biden received the report he requested from his intelligence agencies regarding the origins of the SARS-CoV-2 virus. In May of 2021, Biden tasked the intelligence agencies with generating a report that could, quote, bring us closer to a definitive conclusion as to where the pandemic virus came from. But what the intelligence agencies delivered to the president was deemed inconclusive.
A 90-day review of the origins of COVID-19 appears to be inconclusive. The Washington Post reports that President Biden received a classified report that could not determine whether the coronavirus jumped from an animal to human or escaped from a lab in China. White House officials say lawmakers will be briefed on the report and unclassified details will be made public in the next few days.
For their part, the Chinese government continued to suggest that any interest in the lab leak hypothesis was politicizing the pandemic issue. And they again promoted the idea that if the U.S. pushed for a further investigation into Chinese labs, that the U.S. should then be willing to open up themselves to a WHO investigation, including specifically at Fort Detrick, Maryland, and the University of North Carolina, where Ralph Barrick's lab is.
In a statement on Friday, Joe Biden had accused China of concealing what he called crucial information regarding the origins of COVID-19. It came in the same week the American president received a report from his country's top intelligence services investigating the pandemic's source. Beijing has accused Joe Biden of politicizing an investigation into the origins of COVID-19.
We once again urge the United States not to politicize the whole issue. I would say scapegoating China cannot whitewash the US. If the United States persists in hyping up the lab leak theory, then they should set an example and invite the WHO to investigate the Fort Detrick and the University of North Carolina.
The remarks stem from a reverse theory pushed by Chinese authorities, suggesting that America, in fact, was the birthplace of the virus.
By the end of September 2021, Josh Rogin was writing in the Washington Post that, quote, Biden's promises to press China on allowing a real investigation into the origins of COVID-19 appear to have been quietly shelved. Also in September of 2021, Dr. Filippa Lentzos, who is a senior research fellow at King's College London, where she researches biological threats, spoke at a public conference about the origins of COVID-19. Dr. Lentzos opened her time describing what should have been done at the outset of the pandemic.
Your first step in an investigation is based on traditional epidemiology, asking who is being infected with what virus or bacteria. Part of this process is also looking for potential indicators of unusual sources. So for instance, is the virus unusual for the location? Or is it unusual for the time of year? Is it affecting unusual populations in unusual ways? Are there labs nearby working with any relevant viruses? Now, if a natural source is not identified, or if early evidence indicates a potential lab or research-related source, the search for a natural source should continue, of course, but the investigation should expand to include lab sources. Here, the initial step is to perform a risk assessment of nearby labs to identify first their purpose, then what viruses or bacteria or what unknown samples or suspect samples are being worked on within these labs. Third, what techniques are being used for that work and what sorts of risks that entails. And finally, you want to identify at what biosafety level the work is being done. Now, to do all this, you will need access to all kinds of records, and the more the better. And it should be in the lab's interest to share this because it gives them an opportunity to demonstrate that they operate safely, securely, and responsibly.
Before closing, Dr. Lentzos addressed the question that even Senator Marshall says he is frequently asked. Namely, why does it matter where the virus came from?
Do we actually need an investigation? Isn't the key takeaway from the pandemic origins debate that while we don't know how the virus first spilled over, it could have been either a natural spillover or a spillover resulting from research-related activities. So in order to better prepare for pandemics in the future, we are in any case going to have to address both of these possibilities. Now, I think that argument is right in theory. In theory, it doesn't actually matter what the origin was, because we should be preparing for both instances in the future. In practice, on the other hand, I think it matters a great deal. If it turned out to be a lab leak, or an accident in the course of regular scientific research in the field, for instance, the scientific community would have a very different reaction than if it was a natural spillover. It would bring home the message about the importance of biosafety and biosecurity and responsible research, and of the broader societal responsibility of scientists in carrying out inclusive risk assessments of their work in a much stronger way. I think it would also impact the wider public differently. If it turned out to be a lab leak, it would be a forceful instigator or amplifier of bigger societal debates that need to be had about the sorts of risks that we're willing to take in the name of research. Does the majority of stakeholders, and stakeholders are not just scientists here, feel that lab manipulations of potentially pandemic pathogens is justified? Do the benefits of virus hunting outweigh the risks? Can we even do adequate risk-benefit assessments when both risks and benefits are uncertain?
Dr. Alina Chan also spoke at this conference. She is the molecular biologist who we played a clip of in part 1 of this series, in which she discussed how SARS-CoV-2 did not need to evolve the way SARS-1 did when it first entered the human population. How instead, SARS-CoV-2 was, quote, good to go. At this conference, Dr. Chan spoke about a research paper that had been released in the summer of 2021 titled Animal Sales from Wuhan Wet Markets Immediately Prior to the COVID-19 Pandemic by Xiao et al. The authors of this paper just happened to have been recording which animals were being sold in Wuhan's 17 so-called wet markets for the 2 years prior to the COVID-19 outbreak. As this paper documents over 47,000 animals from across 38 species, proponents of the idea that SARS-CoV-2 spilled over to humans from an animal sold for food at a market leaned on this research as evidence to support their hypothesis. Even the media highlighted the paper with articles such as Bloomberg's August 16th piece that states, quote, a research paper that languished in publishing limbo for a year and a half contains meticulously collected data and photographic evidence supporting scientists' initial hypothesis that the outbreak stemmed from infected wild animals, which prevailed until speculation that SARS-CoV-2 escaped from a nearby lab gained traction. Dr. Chan dug into the numbers to see if they were actually indicative of a likely market-based spillover.
I know that a lot of people cite a recent report in scientific reports that talks about animal sales in Wuhan across 17 stores in Wuhan that sold wild animals. And I'd say that there is a pathway for a virus to naturally emerge in Wuhan, but the numbers are not as robust as what you would see in South China. Remember that in the SARS-1 epidemic, once they had pegged the civet as an intermediate host, they had to slaughter like 10,000 civets in that proximity of Guangzhou. But here, if you look at the numbers from this paper, you look at civets, for example, on average, they saw about 11 civets per month in 29 months across 17 stores being sold with a standard deviation of 8. So this means that sometimes there might have been only 1 or 2 civets being sold per month across 17 stores. So this data, it would be great if you could see the actual numbers, the actual distribution over time. I was told by the authors that in the winter, the sales are really low. So like November, there's very few wild animals being sold. Again, it's just not comparable to the much more robust sale of wild species in South China, where eating wild species is more of a cultural trend.
Dr. Chan then spoke at length about the WHO's research into the Huanan seafood market that back in early 2020, many people just presumed was the site of the spillover of SARS-CoV-2 into people.
So in May of last year, we were finally told that no animals from the market were positive for SARS-2 infection. And actually, the Chinese authorities had known by the end of January 2020 that there were no animals from the market that were positive for SARS-2 infection. They told the OIE in private, and they spent the next few months trying to find animal source of the virus, and they couldn't find anything. By May, the Chinese CDC director came out and told everyone it looks like the market was a victim. The virus had existed long before. A year later, March 2021, they finally came out with this report with the WHO saying that they had tested 80,000 animal samples. More than half were wild animal species. So 41,000-ish were from civets, raccoon dogs, a kind of wildlife samples, so not just chicken and pork. They collected these from 31 provinces between 2018 and 2020. No positive result was reported for any of these samples. The zoonotic origin of SARS-1 was rapidly confirmed. Within 2 months of isolating the virus, they already found widespread circulation in the animal trading community. So I can't go through these in detail, but they found that people who traded in civets, primarily, 73% of them tested positive for SARS antibodies, even though they had not been diagnosed clinically with SARS. So it spoke to widespread circulation exposure of animal traders to SARS-1 at the time. But there was no such evidence for SARS-2.
Dr. Chan even made sure to point out that the wild bats of Hubei province, the province in which Wuhan sits, were widely sampled to find evidence of coronaviruses that could infect people.
Similarly, thousands of bats, maybe tens of thousands of bats in Hubei have been sampled over the years, but none were reported to carry SARS-CoV that could utilize ACE2. Was a SARS-2-like virus likely to naturally emerge in Wuhan? The answer is no.
Finally, Dr. Chan, as a microbiologist, weighed in on what can be determined by examining the genome of SARS-CoV-2.
So if you just look at the genome alone, it can be consistent with natural evolution, but it's difficult for you to know whether it was pre-adapted in the animal and then spilled over in a research-related way, or whether it's natural, but it spilled over many months ago, maybe through a sampling expedition. So there are many ways that a natural virus can still spill over in a research-related way. Even a genetically modified virus may not be possible to distinguish from the genome alone. So this is an ongoing challenge for top scientists even, is that you can't just look at the genome and say, did this ever pass through a lab or cell culture in the lab? We know that from the EcoHealth Alliance's grants that they were characterizing high spillover risk SARS in southern China. So they were deliberately collecting tens of thousands of samples from South China, bringing it back up to Wuhan. They were checking people as well, and working with local clinics and CDCs to collect these samples. Once in the lab, they would test the ability of novel spikes using pseudoviruses or chimerics or synthetic viruses to infect different human cells or human ACE2 expressing cells, as well as animal models such as humanized mice and civets. So we know that a whole range of research activities was happening. They didn't have to just rely on naturally isolated viruses. They could just build it from scratch as long as they had a genome or a spike sequence. We know from a recently FOIA document that these are unpredictable. So when you look at these data, it shows that some of these novel chimeric viruses could grow up to about 10,000 times better than the parent virus that they had isolated or synthesized in the lab. We now know that the WIV was working with 9 closest relatives to SARS-2 when the virus was first detected in December, yet this information was not shared on day 1. It took more than a year for sleuths and internet analysts to collect all of this information. This had been collected from Mojiang mine, where 6 people had sickened with a mysterious pneumonia suspected to be caused by a bat virus with a 50% mortality rate. So this information, again, it was not shared on day 1.
The internet sleuths Dr. Chan is referring to are, of course, members of Drastic. And what they put together is the existence of a clade of coronaviruses called the 7896 clade. In December of 2020, Dr. Shizheng Li gave the keynote lecture for the European Scientific Working Group on Influenza. She titled her lecture, From SARS to COVID-19. In one of the accompanying slides, there was pictured a phylogenetic tree of coronaviruses. And very closely related to SARS-CoV-2 was a list of 8 bat-borne coronaviruses, including one called Bat-CoV RA7896. Prior to this keynote lecture, in a paper published by Alice Latham, a research scientist at the EcoHealth Alliance, there was a reference to the RDRPs, which are a piece of the virus genome, of 8 beta coronaviruses. Where these beta coronaviruses were sampled and when they were sampled was not mentioned in the paper, but a Drastic member who goes by the name Baybar Elephant noticed that the RDRPs of these 8 beta coronaviruses were very similar to those of both RATG13 and SARS-CoV-2. Francisco de Assis, another Drastic member, went looking at the available information on RATG13 and noticed that the file names for several of its amplicons all contained the number 7896. By digging through the file names of sequences uploaded by the Wuhan Institute of Virology, Francisco was able to ascertain that the 8 beta coronaviruses that were mentioned by Dr. Latham in her paper were the 7896 clade, and that they were all sampled in the copper mine in Mojiang, where RATG13 was found. Finally, Drastic member Rosana Segredo found that slide from Dr. Shizheng Li's keynote lecture that pulled it all together. Dr. Jonathan Latham of the Bioscience Resource Project also mentioned the apparent bad faith of the Wuhan Institute of Virology in keeping this clade of viruses from public view from the outset of the pandemic.
So in a very recent paper, the Wuhan virologists explained that they had isolated another closely related lineage but distinct from RATG13 and SARS-2. So they had a collection of viruses that they'd been sitting on for the entire duration of the pandemic. And then when they do publish it, they provide coordinates that don't match the place where they claim to have got the virus from. So this is yet another example of attempting, seemingly, to throw people off the trail of where all these viruses are coming from.
Dr. Jonathan Latham has been further critical of the fact that despite a year and a half of searching, no intermediary host animal between bats and humans has yet been identified.
China has an interest in finding a zoonotic origin for the virus, but they also have an interest in people not knowing that there has been a big search for the origin. There were, we think, around 80,000 animal samples taken, and they were from many different species. We know that one or two virus samples that have been published from China have involved going back to historic samples taken from bats. So we know that the search has not only been for animals existing today, but also in samples taken over the past 10 years or so. So we know that researchers in China have been going back to their archived genetic material of different kinds to find samples that might contain coronaviruses and therefore might offer clues as to a zoonotic origin.
Dr. Latham told me that since the pandemic began, several new coronaviruses that are related to SARS-CoV-2 have been found.
They have turned up viruses from bats, basically all over Asia, that are very closely related to SARS-2. They're more closely related than most of the viruses that we knew about prior to the pandemic. They're not as closely related as you would expect if they were the virus that just jumped into a person in Wuhan. But what they are is a constellation of viruses that are basically siblings of SARS-2 and siblings of RATG13. But these are basically viruses that form a little clique, if you like, a genetically related set of viruses, but they're not actually close enough to be the direct ancestor of the virus. But what they do is they give us clues as to the history of that virus. So we can kind of infer from that they are firstly a co-evolving group, and secondly, that they are basically restricted to Southeast Asia.
Doctors Latham and Wilson did what's called a phylogeographic analysis of the viruses most closely related to SARS-CoV-2 to see what could be gleaned about where the pandemic virus came from.
We took these viruses that are potentially ancestors of SARS-2 or the close cousins of ancestors of SARS-2, and we mapped them. And when you map those genomes, what you find is a very interesting plot in which the viruses which are closest to SARS-2 all map close to south central Yunnan and near to where RATG13 was found, which is a mine in south central Yunnan. And the farthest genetically related viruses are found geographically far away. So those ones are found in Thailand and in the very distant parts of Japan. So what that means is you end up with a correlation. And that correlation basically says that the ancestor of SARS-2 that came from a bat left that bat very close to south central Yunnan where its nearest relative was found. So what we're doing is we're geographically tracing the ancestor of SARS-2 to a very specific place in south central Yunnan. And that has all kinds of implications for zoonotic origins because it means that whatever the ancestor, the bat ancestor of SARS-2 was, it did not come from very far away from that region of China. So it didn't come from Thailand, it didn't come from Malaysia, it didn't come from Japan, it certainly didn't come from Europe. So it narrows down the zoonotic origin possibilities or indeed the lab origin possibilities in a very dramatic way.
Dr. Latham and Wilson's analysis shows that the further away from south-central Yunnan one would go to take samples, the more distantly related the coronaviruses found would be from SARS-CoV-2. It also suggests several things about the possibility of a zoonotic spillover.
It really moves forward the origins discussion because what it says is that SARS-2 came from a bat in or around south-central Yunnan, and it constrains a lot of the possible origins that have already been proposed for a zoonosis. So, for example, people have proposed that the early cases have been found in Europe, and the Chinese authorities tried to argue that the virus came to Wuhan and didn't originate there. And so this is a big deal if you then show that the ancestor of SARS-2 came from south-central Yunnan, because that brings the whole discussion back to China again and says that if there were early cases in Europe, what that means is that China has been offering a misunderstanding of the epidemiology of the virus in Wuhan, maybe hiding early cases that might go back to September or October, or possibly even earlier. What it also does is it takes the zoonotic origin ideas that have been proposed, for example, that the virus hitched a ride on a pangolin, smuggled into the country, and then reached Wuhan, where it infected a person. It makes those theories quite difficult to sustain, because if the virus itself came from south-central Yunnan, then the smuggled animal had to pass through Yunnan. So what that means is that you should be able to search south-central Yunnan and find more closely related bat viruses. You should be able to find the zoonotic intermediate host.
Dr. Latham went on to break down how the phylogeographic analysis can be used to think about SARS-CoV-2 not just as a whole virus, but piece by piece.
There's 4 viruses that are super interesting to figuring out the origin of SARS-2, because these are 4 viruses that between them have the closest genomes to SARS-2. If you look at the spike protein, the virus RATG13 has the closest spike to SARS-2. Presumably, that virus, or one of its very close relatives, is the origin of the SARS-2 spike. If you look at other parts of the genome, there are 3 other viruses between them can basically supply the genome parts that are the super closest related to SARS-2 that we know of. And what's interesting about those 4 viruses is they all come from that part of south-central Yunnan. If you were basically to imagine that SARS-2 originates from a wild virus of some kind, these would be the parents, right? These 4 viruses would be the parents. And it's a human equivalent of, you have a child who has blue eyes and black curly hair and has big feet or whatever. One of the parents is likely to have big feet, and one of the parents has got to have blue eyes, and one of the parents has got to have black curly hair. This is the virus equivalent of doing that. You identify a grandparent with big feet and a grandparent with blue eyes and a grandparent with black curly hair. You're not saying that each one of those is exclusively the parent, but you're saying that those are the genetic subparts that you need to build the offspring.
Back in part 2 of this series, Dr. Latham explained his hypothesis that SARS-CoV-2, or its direct progenitor, evolved in the body of one of the sick miners who were clearing bat guano from the mine in Mojang. I asked Dr. Latham how this phylogeographic analysis affected that hypothesis.
We proposed that the SARS-2 virus originates from blood samples that were taken from miners that became sick as part of an apparent coronavirus outbreak in 2012. These 6 miners became sick, and 3 of them died, and they were diagnosed with a probable coronavirus infection. We know blood samples from those miners were taken and sent to the Wuhan Institute of Virology. So our hypothesis basically is that SARS-2 evolved from those miners and was extracted from those blood samples in the Wuhan Institute of Virology, and then either that itself was SARS-2, or some manipulations and so on and so forth were done with the virus. And so what's super interesting is that the phylogeographic analysis supports the idea that the virus came from this mine because that is the place where the most closely related viruses are. In order for the Mojang theory to be correct, all the different parts of SARS-2 has to come from the mine. These other viruses that are also putative parents have to have been in the mine at the same time in order to contribute to the development of SARS-2 inside the miners' bodies. But it does make it a very plausible scenario because all these closely related viruses come from very close to the mine. The most similar genome parts that we need to assemble SARS-2 are all found in that place.
As we were finishing this episode, a scientific paper was released with the very wordy title, Coronaviruses with a SARS-CoV-2-like receptor binding domain allowing ACE2-mediated entry into human cells isolated from bats of Indo-Chinese Peninsula. In this paper, the authors detail that after sampling bats in Laos, which borders Yunnan, they found 3 viruses that are like RATG13, slightly more than 96% similar to SARS-CoV-2. Various media outlets claimed this discovery as a victory for those who promote a zoonotic origin for SARS-CoV-2. Science magazine hosted an online panel on September 30th of 2021, in which the moderator mentioned these newly discovered viruses.
A paper came out a couple of weeks ago from a research group in Laos, working with the Pasteur Institute there and in France, that found 3 new isolates that are closer, but they're also closer, most importantly, in the way that the virus actually binds to human cells. And it almost perfectly matches in these new bat viruses the SARS-CoV-2 receptor binding domain.
One of the panelists, Dr. Jesse Bloom, who is an evolutionary biologist at the Fred Hutchinson Cancer Research Center, as well as Bloom Lab, was asked by the panel moderator if the discovery of these viruses in Laos altered his perspective regarding the origins of SARS-CoV-2.
I've spoken with several researchers who said to me, this furthers the case of natural origin because you don't have to imagine that a laboratory created this. Jesse, does that alter your thinking at all? Does this have any influence on how you tilt things, natural versus lab origin?
Honestly, not really. So I think that due to studies like the one you're describing, we now have an increasingly good idea of there are viruses that could be the deep ancestors of SARS-CoV-2 that have been identified in Yunnan, like this RATG13 virus, have been identified in Laos, like the virus called Banal 2052 that you're just referring to. And then we know what SARS-CoV-2 is, and the question is, how did we get from that deeper ancestor to SARS-CoV-2? The plausible ways that this virus could come from a lab have to do with the lab collecting SARS-related coronaviruses that they think are going to have the potential to jump to humans, like be able to bind to human ACE2 and things like that. And then, either have an accident with one of these viruses they've collected or in the process of slightly modifying that virus. Those are the plausible versions of the lab accident theory. Obviously, those are contingent on those natural viruses existing out there in the first place. So I think the question is not, are there bat SARS-like viruses related to SARS-CoV-2? The answer is clearly yes, and I think it's great that we're getting a better understanding of what those viruses are. But the question is, how did those bat SARS-related coronaviruses that are the direct ancestors of SARS-CoV-2 get to Wuhan and start spreading in humans? And that question, I don't think we're further along.
I sent an email to Dr. Jonathan Latham to ask him about these viruses found in Laotian bats, and he wrote back to me, quote, "The paper itself states the closest SARS-CoV-2 bat-borne genome still remains RATG13, and this is indicated also in the phylogeny they generate. Nevertheless, this is an important analysis. By finding a receptor binding domain that is virtually identical to that of SARS-2 in a bat virus, what this paper does is show again that the key sequences that compose SARS-2 come from near to the mine. In this case, a sampling site 500 kilometers from the mine, which is quite local in terms of the Asian distribution of the SARS-2 clade. This broadly confirms our overall analysis and supports the Mojang minor passage theory. Dr. Latham also pointed out to me that Banal 52 and the other viruses found in the bats in Laos do not have a furin cleavage site. We also know, thanks to the work of drastic researchers, that the Wuhan Institute of Virology and EcoHealth Alliance had been sampling bats in Laos for coronaviruses for years prior to the pandemic. If when the COVID-19 pandemic began, the Wuhan Institute of Virology was in possession of not just the closest known virus to SARS-CoV-2, RATG13, but also the 8 other most closely related viruses known at the time, the 7896 clade, and if all of them came from the Mojiang mine in Yunnan, why was this information kept from the rest of the world? Why did members of Drastic, working in their free time and posting their findings to Twitter, have to force this information into the light? And what other information was available in the files and records of the Wuhan Institute of Virology and in the offices of the EcoHealth Alliance? Join us for Part 6 of Origins, Birth of a Pandemic. Origins was written, reported, and edited by me, John Duffy. Our producer was Ray Novoselsky. Field audio recording by Kristen Turo. Original score by Zachary Walter. Origins is made without sponsors and is paid for by listeners like you. If you can, please make a donation to our Patreon account at patreon.com/double*media, or simply donate via Cash App at DA Media 2021. Also, please rate and review this show on Apple Podcasts and share and suggest it to friends who you think would find it interesting. Thank you for listening.